more on the resident microflora of the human body. Let's focus on the gastrointestinal tract. Okay, so usually we don't have anything in the stomach. Um, we don't expect microbes to really thrive in our stomach because of the acidity of the stomach acid. Um, in the duodenal and jejunal area of the small intestine, most of the microbes that dominate, or the microbes that dominate as normal flora, are the enterococci and lactobacilli. And in the large intestine, okay, the colon area, so a more diverse group are found, right? So from coccoids to rod shape enterobacters, okay, uh, as well as streptococcus and staphylococcus. So these are normal. Some representatives of these genera are known as normal microflora, or are identified as resident microflora. In the event of in the event of okay, so in the event of GI sterilization. So what is this example? What example can we cite? So treatment, antibiotic treatment, drug treatment. So when antibiotics are consumed by the host, these antibiotics usually lead to the targeting of all kinds of microbes found in the GI tract, especially in the intestines. Okay, so they can clear the normal microflora. They can eliminate these microflora. They also target these microflora in terms of mechanism of action. So we expect the normal microflora to also change in terms of their population structure, their abundance. Okay, in such instances, those representatives that are resistant to the drug, the antibiotic that was used, can actually take over, can dominate. No, those antibiotic resistant groups or species um, may dominate the area after sterilization. No, okay. In terms of other functions or associated functions with these GI microflora, bacteria in the large intestine, especially for in the colon bacteria, actually produce gases, carbon monoxide, nitrogen, methane, and hydrogen, okay, and also associated with odor compounds, okay. So to sum it up, this normal flora usually are not important for our survival. They're not essential for us to live. But of course, since they are omnipresent, they're found everywhere. The what happened is we as a group of animals develop this intimate relationship with them. Okay, they're normally, as mentioned, they're normally harmless, provided that they are not found or they are not introduced in areas such as the blood, the lymph, and other internal organs such as the brain tissue, the nervous tissue proper. Okay? It's important that we also know their characteristics. We also know their distribution because their presence influence whether there is disease caused by a truly pathogenic microbe. Okay. Now, a question I'd like to further talk about is how did we as animals get colonized by by microbes, both mostly for the case of the resident microflora. No, so we review that inside our mother's womb, in utero, we are essentially sterile. We're germ free. We don't have any kind of microbe in our bodily tissues yet. Okay, so generally, while inside the mother's womb, the animal, the human, um, is sterile, doesn't is not associated with any other microbe or with any kind of microbe at all. No? However, there are some exceptions to that. Um, there are microbial agents that can be passed from mother to child, from mother to the offspring. So these are known as the torch microbes, which can be passed during pregnancy, during delivery, or even after birth. So these include toxoplasma, rubella. There are other infections um, that can be passed via mother to child route, no, the in utero route. As a good example, we have viruses, cytomegaloviruses, herpes. Also, as far as I know, HIV can be transmitted from mother to child. Okay. All right. So after being born, there are several sources of normal microflora that will try to colonize the newborn's body. No? And some of these include the following. During passage through the birth canal, the newborn will most likely have its first exposure to microbial agents. Microbial agents found in the female reproductive tract, in the vaginal canal. Okay, they are exposed to the skin, or the skin of the newborn is exposed to these microbial agents. Also, by contact with feeding materials, um, pathogens, and not pathogens, microbes can start exploring, colonizing the newborn's body. Contact with measurements, such as whenever the newborn is checked by the medical. Okay, so instruments such as a stethoscope can carry microbial organisms that can be passed to the newborn. Contact with other people, definitely. That's why it's very important that we don't really touch, or we don't really hold um, newborns that are not part of our household. So it's not a good practice to, um, be it's not a good practice to actually hold another person's baby, because you don't know what kind of microbe you will be transferring to that newborn. And definitely during nursing, no, during breastfeeding, the mother can introduce her normal microflora to the newborn baby. Okay? Uh, the establishment of normal microflora in the newborn usually starts at birth. As an example, lactobacilli that can be found in the vaginal canal. It's one of the first to establish themselves in the newborn's skin, or in the newborn's body. And eventually, they predominate in the intestine, in the gastrointestinal tract. So at the start of birth, or at the time of birth, we expect that to coincide with the first exposure, no, the first contact to microbes. More microorganisms later on will be introduced during breathing and feeding events. And other harmless microbes colonize other body parts, such as skin and mucous membranes, as we become exposed to the external environment, as the newborn becomes exposed in his or her environment, in his or her external environment. As days pass by, more microbes, typically the harmless ones, will begin to colonize the newborn's body parts. Okay. So as an example here of an introduction to foreign areas in the host. Okay, so our normal microflora in the oral cavity, Streptococci viridans, usually found in the mouth, no, and the upper respiratory tract. As an example, we have Streptococcus mutans. in the mouth, in the oral cavity, you expect them to be resident microflora. They don't cause any harmful sequelae or harmful infection. But if they are introduced to the blood, let's say to the cardiovascular system, they may cause inflammation of the heart. No? They may cause endocarditis when introduced into the bloodstream. So we expect that in the absence or when in the instance of usual or in the instance of a tissue breach, meaning the microbe has been introduced to a foreign area in the human body where it is not supposed to be or where it's not supposed to thrive, then infection may happen. Okay. Another example of now more internally. So let's look into bacteroids that are normal microflora of the colon. So these bacteroids found in the gastrointestinal tract, in the intestines, are elongated, not morphologically, most of them are elongated cells. You can, as you can see here in this photomicrograph. Okay, if they spread to adjacent tissues such as the bloodstream or the other bloodstream or the what do you call this, the appendix for one, so this small structure here, if they spread there, they inhabit and some instances or predisposing factors cause them to go amok, to start a cycle of pathogenesis. So they become harmful. Okay, they can cause infection. So for the case of bacteroids that can lodge into the appendix, they can cause inflammation of the tissue or the inflammation of the part. That's appendicitis. If these bacteroids become or if these bacteroids gain access to the bloodstream, and they can cause bacteremia, they can also cause inflammation of cardiac tissue. They can cause endocarditis if later on they are found in the heart, they are found in the cardiovascular or the cardiac muscle tissues. If they begin or if they gain access to that area, they can cause infection. Okay? Now, some examples of predisposing factors that enhance or that lead to unfortunate tissue breach of these pathogens. An example is the use of oral contraceptives, which can disrupt the normal acidity and moisture of the GI tract. In such instances, the microbes may behave differently. The normal microflora will behave differently and may explore alternative spaces where they can try to where they will try to thrive or to survive. No, other examples include diabetes, which is a system which is known to be associated with systemic problems, with systemic issues. The immune function of individuals with diabetes is usually downgraded, or it's not at its optimal. As such, the body may be at risk of having opportunistic infections caused by normal microflora. Okay? Now, overall, okay, we expect that truly endogenous or the normal resident microflora, they are beneficial. No? They're known to actually more. They're found. There are a lot of findings on how they benefit the human body. No? Overall, they can provide good things. No? They can make the human body. They can contribute to the survival of the human host. Again, and in that sense, they're harmless. Okay, but as specific examples, no, I think I've mentioned in our on-site lecture, no, that one of the basic reasons why we study microbes is because they benefit us, no. There are a lot of things that they can produce, such as vitamins and essential growth factors that we can consume, that we can use during metabolism, right? And they also participate in the metabolism of food products, and also the absorption of the nutrients derived from such metabolism. A good example here are microbes that can degrade cellulose. So for those who are strictly on a vegetarian diet, okay, you expect that their gastrointestinal tract, the microflora, will be dominated by normal groups or normal species that have the capacity to degrade cellulose. So in such sense, and in that example, we appreciate how the normal microflora participate in the metabolism of food products. Okay. Also, one other benefits include the protection against other infections, mostly by highly virulent microorganisms. So, as normal microflora are known to dominate the specific body parts, specific body tissues, any pathogen or any potentially pathogenic microbe or species, it will be outnumbered, no, by the normal microflora, and they have slimmer chances of being able to establish themselves in a normal healthy host. Okay, competition for nutrients and receptor sites on the host cell. So also one expected outcome caused by or associated with the presence of these microbial agents is sorry this microbial normal microflora in the body. Okay. Other benefits or other advantages, they can stimulate immune response, uh, growth factors that are associated with metabolic pathways for the production of essential immune factors, and for the maturation of some essential immune cells. Okay, they can also produce normal microflora can also produce bacteriocins and other inhibitory substances. So as discussed last time, I think I remember talking about how microbes can also be studied because of the bioproducts that they produce. So in the context of normal microflora in our body, so some of them can produce inhibitory substances in their attempt to, of course, survive. No, it's definitely that there are a number of interactions happening inside the human body and even on the external surfaces. So you expect that each species of normal microflora will try to optimize its environment in order for it to thrive. So in such cases, it's possible that they produce substances to inhibit the growth of others. And with that, no, we achieve this so-called balance or equilibrated population structure of normal microflora. The interactions that are not so beneficial and harmful ones definitely include disease, no, or infection. Disease is said to be an abnormal state where a part or all of the body is not properly adjusted in the presence of a microbial agent that is also trying to overcome the response of the human host. Okay, so essentially the from this point forward, all our discussions will cover the harmful effects of microbial organisms from different groups, different categories, in the context of infection. So, ano na, no? Moving forward, we'll look into the medical microbiology of the microbial agents. So, all the other, the hybrid between infection control and also disease development. Okay? So that ends our module number 2 on the introduction to medical microbiology.